Good evening. Welcome to a lecture on biotechnology and sustainable agriculture. Now, this topic has been selected or designed both from PT point of view and the men's. The facts here are to be discussed. Will be useful for those who aspire for PT and thereafter for the men's exam. So biotechnology, the term is not unknown to you. Biotechnology means a technology that uses biology. Isn't it? So the use of biology for the mankind and to make things that are beneficial for the mankind. This is a biotechnology. And here is genetic engineering, so-called recombinant DNA technology. So, before switching over to how this biotechnology to promote sustainable agriculture, I would like to discuss some points, say one by one. So the first point, the microorganisms, animal or plant cells or their components to generate products and services beneficial to human beings is called biotechnology. Unknowingly, humans have been using microorganisms for thousands of years to get products like curd, wine, vinegar, bread, etc. Next, macroorganisms were first used to produce some organic compounds like citric acid following the First World War. And the duration of the First World War is, what is the duration?
1940 to 1980.
1914 to 1918.
Yes.
Okay, 1914 to 1918, isn't it? Next, come to know the transgenic plants. A gene that is transferred into an organism by genetic engineering. And just I told you, genetic engineering is also known as recombinant DNA technology. It's called transgene. An organism that contains and expresses a transgene is known as transgenic organism. Keep a smiling, dear all. Your smile will catalyze me to work more and more with utmost concentration and no less focus as you give preparation to your exam. So come back. Genetic engineering allows or permits the isolation of desired gene from any organism and its transfer and expression into the organism of your choice. Next, come to transgenic macroorganisms are produced to get novel pharmaceuticals proteins, and the proteins produced by transgene are called recombinant proteins. For instance, human insulin is commercially, that is on large scale, produced from a transgenic Escherichia coli strain that contains and expresses the human insulin gene. So now come to the transgenic crops. Transgenic crops are also known as genetically modified crops. Okay? Advantages of this technique. Number one, any gene from any organism or a gene synthesized chemically can be used for a transfer. First point. Second point, the change in genotype can be precisely controlled. It's because only the transgene is added into the crop genome. Next fact, when a transgene is introduced into the genome of an organism, it can achieve one of the following. Here are the points. To repeat, when a transgene is introduced into the genome of an organism, it can achieve one of the following. First point, produces a protein that is the product in which we are interested. Second point, produces a protein that on its own produces the desired phenotype. Right? Third, modifies an existing biosynthetic pathway so that a new end product is obtained. And fourth one, prevents the expression of an existing gene. This is all about the new technique. GM crops are already in cultivation. You may be knowing it. Just to repeat, genetically modified crops, GM crops are already in cultivation in United States of America, Europe, and in many other nations. In our country, some insect-resistant cotton varieties expressing cry genes have reached the farmer's field. Now come to GM food. Food prepared from the produce of genetically modified means transgenic. That is the food that we get from the transgenic crops. If the crops are transgenic, naturally the food will be what? Transgenic. So now see, so food prepared from the produce of genetically modified, that is a transgenic. GM food differs from the conventional developed varieties in these aspects. There are two kinds of food. One is the genetically modified, so-called the transgenic GM food, and next one is the conventional. How the two are different. Number one, understand the point? So it contains the protein produced by the transgene in question. I am talking to what? GM food. These facts are for the GM food. These facts are for the GM food. It contains the, and I mean the GM crops or the GM food contains the protein produced by the transgene in question. For instance, cry protein in the case of insect-resistant varieties. And second point, GM food or the crops contain the enzyme produced by the antibiotic resistant gene that was used during gene transfer by genetic engineering. And third point, finally, it contains the antibiotic resistant gene itself. Now, concerns and GM crops, so-called the genetically modified crops of the food. What are the concerns over this? Over the genetically modified crops of the food. What are the issues to be taken into account? Number one, transgenic product may cause, very important point, transgenic product may cause toxicity and or product allergies. Second point, the enzyme produced by the antibiotic resistant gene could cause allergies since it is a foreign protein. Finally, the bacteria present in the alimentary canal of the human would pick up the antibiotic resistance gene that is present in the GM food. These bacteria become resistant. These bacteria become resistant to the concerned antibiotic. So these bacteria would be difficult to manage. However, efforts are being made to use other genes in place of antibiotic resistant genes. Toxic and allergenic action of the transgene product to be adequately addressed. Thanks for giving me a patient listening. Thank you.